Hello students, welcome to UPG Pathshala. I am Dr. P.M. Reddy from the Energy and Resources Institute. Today we are going to discuss about a module on the detection, characterization, expression of transformants. In this, we discuss selectable markers, reporter genes, target stability, and gene silencing under the paper Plant Biotechnology and Crop Improvement. The learning objectives include to familiarize with the strategies adopted to selectively encourage regeneration and development of transgenic plants, to understand the key features of selection markers used in raising transgenic plants, to comprehend usefulness of reporter systems in deciphering molecular, biological, and physiological process in plants, to familiarize with the strategies used to silence gene expression in plants to help decipher gene function. Among the reporter gene system, the selectable markers are the very important ones. And the selectable marker genes for screening transgenics is extremely important to make the transgenic plants. Marker genes can be categorized into two groups, selectable markers and reporter genes, or scoreable markers or screenable markers. A selectable marker is a gene introduced into a cell that confers a trait suitable for artificial selection. There's a picture given here is a selectable marker, which is shown here is HPT. It's hooked up to a promoter. Normally, for a selectable markers, a constitutive promoter is used. And then when the promoter is expressed, a particular gene is expressed and the enzyme is produced to decompose or neutralize a particular antibiotic or herbicide or antimetabolite and so on. Advantages of marker genes: they are easy to select transformants. It does not require DNA extraction, electrophores, and other methods to select the transformants. Different types of selectable markers include antibiotic resistant markers, antimetabolite markers, herbicide resistance markers, etc. There are different types of selectable markers have been used to select the transgenic plants, and these markers include positive selectable markers and for negative selection. And most of the selectable markers which have been used for positive selection, they include first category, antibiotic resistant markers. There are several genes which have been used to incorporate into plant, which give resistance to plant against certain toxic concentrations of antibiotics, like NPT2, which codes for neomycin phosphorotransferase. This is being incorporated to confer resistance against kanamycin in plants. This particular enzyme, neomycin phosphorotransferase, detoxifies kanamycin, neomycin, poromycin, etc. Another gene very commonly used is HPT, which produces hygromycin phosphorotransferase. This enzyme detoxifies the antibiotic hygromycin, which is used in the selection medium. There's one more is AAC3, which codes for 3N-aminoglycoside acetyltransferase, which detoxifies gentamicin. And similarly, AATA, streptomycin phosphorotransferase. This particular enzyme detoxifies streptomycin antibiotic. There are other selectable markers which rely on herbicide resistance. They confer herbicide resistance to plant cells. These markers include BAR, which codes for phosphinothrycin acetyltransferase. This enzyme detoxifies L-phosphinothrycin, This is commercially known as Biolafos, which is abbreviated as a PPT. There's another gene which confers herbicide resistance is ROA, which codes for 5-enol pyruvyl shikimate 3-phosphate synthase, EPSP synthase. This particular enzyme detoxifies and makes plants to insensitive to glyphosate, that is commercially available herbicide which is commonly known as Roundup. This is one of the very important genes to confer resistance against glyphosate herbicide. Another gene is CSR1-1, which codes for istolactate synthase. It's a mutant form of it, which neutralizes sulfonylurea and makes plant insensitive to this antibiotic. And once herbicide. Once this herbicide is incorporated in the medium, if they express this particular gene, they are able to grow in the presence of this herbicide. Another is BXN, bromocynyl nitrulase, which detoxifies bromocynyl, and this is also used as a herbicide-resistant marker. A third category is antimetabolite selectable marker. There are some metabolites which are being neutralized by certain enzymes. A gene which is known as DHFR, which codes for dihydrofolate reductase, once it's expressed, it makes plant insensitive to methotrexate. And another is DAO1 gene. This gene codes for amino acid oxidase, and expression of this enzyme, D-amino acid oxidase, can convert alanine, serine, D-serine, D-alanine, which are toxic to plant cells. They are detoxified, then produced as a non-toxic substrates are produced, and in the presence of D-alanine and D-serine, when this enzyme acts, the plants can grow. This is a sort of a positive selection like earlier ones. Same enzyme also acts upon D-isoleucine, D-valine, and these amino acids, dextroamino acids, when acted upon by amino acid oxidase, can be converted into toxic substances. So in the presence of these amino acids, the toxic substances are produced due to the activity of this particular enzyme. So the plant cells will be killed because of this. So this is known as a negative selection. This table, which is depicted here, gives a selected list of selectable marker genes used for gene transfer in plants. Upon expression of these genes, resistance is created against different types of compounds. All of these genes are positive, that's for positive dominant selection. Recently, one particular gene is used, which is isolated from Pseudomonas strugeri. It's a PTXD. This is isomer-specific 2-hydroxy acid NAD-dependent dehydrogenase. When this is expressed in the plant is able to utilize phosphite and survive in the medium containing phosphite. Normally, plants cannot utilize phosphite. Plants can only use phosphate. So, in the presence of this enzyme, phosphite is converted into phosphate, and so that plant cells can use in the culture and can grow. So, this is also a positive selection marker. And the second part of this lecture is talking about markers, reporter genes. Reporter genes are nucleic acid sequences encoding easily detectable proteins. Here also, as you have seen earlier with selectable markers, reporter gene can be hooked up to a promoter. And these promoters here in this case can be tissue-specific promoters or inducible promoters or constitutive promoters depending on your need. Different types of promoters are used. The reporter genes here are normally, which are used, are LUC gene or a GFP gene or a DS-RED to produce a sort of a bioluminescence in case of LUG gene, which codes for luciferase, or a fluorescence from the proteins GFP or RFP. Several different types of proteins are being used, and these are very important to study make the things easier for people to study the cellular processes. Reported assays are generally performed to measure gene expression or transcriptional activity, or assay the activity of transcriptional factors, or promoter activity, or conformation of transgenics, because they can be visualized very easily. There's a wide choice of reporter genes available. Among the reporter genes, one of the most common ones, which was extensively used earlier, is CAT, Chloramphenicol Acetyltransferase. This gene, this particular enzyme, transfers radioactive acetyl groups to chloramphenicol, thereby making chloramphenicol ineffective to kill plant cells. This particular enzyme activity, it can be detected by thin layer chromatography or autoradiography when you are using radioactive compounds, or ELISA, the enzymatic reaction. This activity of CAT gene can be visualized. Another reporter gene is B-Galactosidase. This is coded by LAC-C. This hydrolyzes several galactosides into galactose. And also we can use certain compounds upon cleavage by galactosidase. They yield a blue product which can be visualized on the microscope, the naked eye. And another reporter gene, GUS. This codes for beta-glucuronidase enzyme, which hydrolyzes colorless substance like X-Gluc into a blue product upon its activity, and which can be detected very easily. X-Gal and X-Gluc, these are used destructive methods. You have to use a stain and the cells are being killed to visualize that color. There are some reporter genes which can be used, which are not destructive. They can easily seen under the microscope, but cells will be alive. One example is Luciferase, coded by the gene LUC gene. It oxidizes luciferin-emitting photons, bioluminescence. It reacts with the luciferin to produce light. This can be assayed by luminometer or photon counting camera and so on. You have a green fluorescent protein, red fluorescent protein, DS-RED, and so on, which we'll be talking in subsequent slides. CAT gene, which codes for chloramphenicol acetyltransferase. This is the first reporter gene used to monitor transcriptional activity of promoters. This CAT gene is derived from the bacteria. This gene codes for acetyl, as I mentioned, chloramphenicol acetyltransferase, which has the ability to transfer acetyl groups from acetyl-CoA to chloramphenicol, thereby detoxifying this particular antibiotic. Reaction of acetylation can be assayed or quantified by monitoring acetylation of radio-labeled substrate C14 chloramphenicol, or by ELISA, which is a non-radioactive assay. Assaying with the C14 chloramphenicol is a radioactive assay, which depicts ELISA assay. In this, antibodies of CAT protein are being reacted, and which is being acted upon by anti-CAT, which is conjugated to DIG. This particular DIG can be reacted with ABTS substrate to produce a sort of a fluorescence, which can be visualized by subjecting the cells or blood to an X-ray film. This is or it can be read by ELISA plate reader. The fluorescence can be read. This is the assay which is commonly used to measure cat activity. Beta-Galactosidase, this particular enzyme, is encoded by LAG-C, which hydrolyzes, as I already mentioned, beta-galactosides sugar into monosaccharides. Expression of the LAG operon is induced by lactose, and also it is induced by lactose analogs like IPTG, isopropyl BD1 thiogalactopyranoside. When expressed, this beta-galactosidase enzyme can break down dye linked to substrate called X-gal. Chemical name is 5-bromo-4-chloro-3-indolyl beta-D-galactopyranoside. When the X-gal is cleaved, it converts to galactose and also an insoluble blue pigment, which is 4-chloro-3-bromo-indigo. This blue color can be easily visualized. This is particular, though this is a very popularly used reporter system for bacteria, it is rarely used in plants because this particular enzyme is also active, present in plants, which can produce a blue product, endogenous enzyme. So this is normally not used that much. There are different types of assays, either colorimetric, fluorescent, chemiluminescence, or just visual detection of a blue product. Which is the reporter system, which is very commonly used in plants, is GUS reporter gene system. This particular GUS protein, that is beta-D glucuronidase enzyme, is coded by a gene called as UIDA or GUS-A gene from E. coli. When this particular gene hooked up to promoter is transferred to plant, and the promoter, when it's active, this particular enzyme is produced, glucuronidase. This enzyme may be detected by using a compound known as X-gluc, which forms an intense blue product after cleavage by beta-glucuronidase. There's a reaction similar to beta-galactosidase. Here you can see in this picture at the lower bottom, you have plantlets stained blue, which is due to the activity of GUS gene. And a cartoon is presented here. A promoter hooked up to GUS gene can be expressed in the plants to produce a blue pigment in the plant upon addition of X-gluc. And the reaction is also shown here, how the X-gluc is converted to a blue product. Another reporter gene system is very commonly used is Luciferase system. This is the enzyme coded by Lux gene or Luc gene isolated from firefly. This gene codes for enzyme luciferase, produces blue light by ATP-dependent oxidation of luciferin. The reaction is shown here in this slide. And on the right side, there is the picture is given, which shows the fluorescence bioluminescence produced due to the activity of this luciferase enzyme on luciferin, thereby emitting the bioluminescence. Bioluminescence is a process in which living organisms convert chemical energy to light. The light results from oxidation of an organic substrate, as I already mentioned, luciferin, catalyzed by an enzyme called luciferase. Bioluminescence or light emission is determined by luminometer. There's another very popular reporter system. Is a visible marker is using green fluorescent protein, which is in short known as a GFP, commonly known as GFP. GFP gene is isolated from jellyfish, Aquaria victoria. This is one of the very important genes which are used in cell biological experiments. This gene can be cloned behind any promoter, whether it's a tissue-inducible promoter or a temperature-inducible promoter or a conditional promoter or constitutive promoter, whatever it is. When it is hooked up and fused to a promoter, and when the promoter is activated inside the cell, the protein which is produced is a green fluorescent protein, which fluoresces green when you shine blue light of the wavelength of 360 to 380 or 440 to 480. Its emission is in the green wavelength 520. Several GFP variants, they have a different fluorescence emission spectra, and as shown in the picture here, different tubes emitting different types of fluorescence, green, yellow, red, and so on. These are all coming from same protein but of different variants. Gene is mutated to produce different colors. There's on the right side, there is a picture shown different animal systems expressing green fluorescent protein. You can visualize this. This is also very commonly used for transforming plants to visualize the promoter activity. Uses of green fluorescent protein as a reporter molecule to monitor gene expression. Transgenic organism made with the GFP coding sequence under the transcriptional control of a promoter belonging to gene of interest. You can take any promoter and fuse this particular gene and can be used to visualize the expression of the promoter. Here it's depicted a gene A promoter is taken and cloned, GFP is cloned downstream of that and used for transformation of plant or animal cells. You can see in whichever cell this protein is expressed, they fluoresce green color. Besides using GFP protein to visualize the activity of promoter or activity of gene expression, we can also use this particular protein to tag another native protein to localize where this protein is being targeted in the cell. You can see here, in this what is done is, any promoter can be used here to activate this particular gene. For example, a gene is, you want to see the product of this gene, the protein where it is targeted. So you take that gene along with its promoter, and downstream of that, you can clone GFP gene. You will create a fusion protein. When the gene promoter is activated, the native protein as well as GFP, which are fused together, they are expressed. And when you observe the cells which express these fusion gene under the microscope, under appropriate light conditions, you will visualize the places where this protein is targeted. In other words, a fusion protein can indicate where actually inside the cell this protein is targeted and localized. That's how the tagging of the protein can be used. The fluorescent protein can be used to visualize subcellular localization of the protein. So far we have completed the discussion about marker genes and reporter gene systems. Now we come to another important aspect in the plant biology. To study the functions of gene is through gene silencing. When a particular gene is silenced, that means a particular phenotype is not developed, is not formed. You can visualize very easily and what type this gene and its phenotype very clearly. The phenotype can be biochemical, morphological, whatever it is, or any other process. Gene silence can be at different stages. Transcriptional gene silencing, which is called a TGS, when the promoters are silenced due to hypermethylation, or post-transcriptional gene silencing, that is, gene is transcribed and mRNA is either methylated or degraded, that is post-transcriptional gene silencing. One of the common methods which is used to study post-transcriptional gene silencing is through antisense or RNAi-mediated gene silencing. There are several other gene silencing processes there. For example, post-translational gene silencing. When the transcript is translated into protein and protein is degraded, so the gene phenotype is not produced. That's also a sort of a gene silencing. There are different types of gene silencing, as I mentioned here. There are different terminologies are used for this PTGS. RNAi, that is RNA interference, or microRNA for mRNA silencing, or in case of fungi, it is known as quelling. And most common ones are microRNA or antisense RNA, RNAi-mediated silencing. these microRNAs or siRNAs can spread throughout the plant to silence the gene in all different types of tissues, wherever that promoter is, wherever the transcript is present. What is RNA interference or post-transcription gene silencing, and how it can be achieved? Double-strand RNA needs to be directed against an exon, not an intron, in order to be effective. Homology of double-stranded RNA and the target sequence or mRNA is required. They should be complementary for silencing to be achieved. Double-stranded RNA triggers cleavage of homologous mRNA. Targeted mRNA is lost after cleavage or degradation. And the effect is non-stoichiometric. Small amounts of double-stranded RNA can wipe out an excess of mRNA very easily, point to an enzymatic mechanism which are described in the due course of this lecture. Production of double-stranded RNA by various means, transcription of inverted repeats, which is described in the following slide, how the vectors are being constructed, or silencing can also be achieved by using viral RNAs, viral replication, and transcription of RNA by RNA-dependent RNA polymerase and so on. Thus, special type of vectors which have been developed to study post-transcription gene silencing, which are known as RNAi vectors. RNAi technology is a very powerful technology to study gene function and effects of gene silencing in any plant or animal or any other system. Here on the left side, you have a panel in which different types of RNAi vectors, how they are created. You can take a coding gene, in this case what shown is the Gus gene UID gene. On both sides you hook up with a constitutive promoter. That means both the sense and antisense strands of UID gene are transcribed, and these ones are transcribed, and these transcripts can hybridize with one another, forming a double-stranded DNA. And that double-stranded DNA can be subjected to cleavage by a protein system known as a dicer system, RNase 3 system, family protein. And this, once the DNA is cleaved into smaller fragments of 21 to 25 base pairs, they are being recognized by agar-knot proteins and form a complex. This will guide these small RNA fragments to double-strand RNA fragments to RISC complex, which is RNA-induced silencing complex, where this double-stranded small RNA molecule, one of the strands is degenerated, and target RNA, that is a native RNA produced by the plant cells in our case. Wherever there's a complementary sequence for the target RNA, this is the siRNA bind to that, and the RISC will, the double-stranded transcript, the structure hybrid form, it is being degraded by endogenous cell enzymes, thereby silencing the gene expression. When the transcript is not there, gene expression is not there. Another way to make a vector is, which is again shown at the bottom of the left panel, using a single promoter, in this case 35S promoter. UID gene is cloned both in sense direction and anti-sense direction with a spacer in between. When this particular gene is expressed and both sense and anti-sense transcripts are formed, which are joined by a spacer, they form the hairpin RNA structure. Complementary sequences come together, form a hairpin, and they'll be degraded as I have described earlier, which is depicted on the right side of this particular slide, using dicer, agoproteins, and RISC, and how the hybrid RNA molecule is degraded by the endogenous enzymes. Another system which is used to silence a gene is virus-induced gene silencing. This is sort of another form of post-transcriptional gene silencing. Here, viral genome is used to silence the genes. As on the left side of this slide is shown a vector, two vectors, which are TRV1, TRV2 from tobacco rattle virus. This is a bipartite genome, and one of the molecules of this RNA, one of the molecules is codes for replicase, mobility protein, and other 16 Dalton protein, which are all essential genes. And the TRV2 code protein, which is essential, and the two other proteins, which are structural proteins, non-essential. So a gene of interest can be cloned here by replacing non-structural proteins. And when these two molecules has a cDNA hooked up to promoter and expressed in the plants, and it is being transcribed and mRNA is produced, and the internal promoters of RNA will continuously go on transcribing using RNA-dependent RNA polymerase produced by the TRV1 vector to continuously transcribe the RNA molecule. Once it's transcribed, this goes and anneals with the complementary RNA sequences of the endogenous transcripts of endogenous gene. When this transcript form a double strand, they'll be immediately digested or cleaved by DISA, the same mechanism as I described earlier. And these small interfering RNAs, double-strand RNAs, are assembled in RISC, where one of the strands is degraded, and RISC brings the mRNA target, and complementary sequences are annealed. And once the complementary sequences are annealed, the mRNA target is cleaved by the protein degradation proteins of the cell machinery. What is the DISA? DISA triggers double-stranded RNA, triggers processing of double-strand RNAs by enzyme RNase 3 families, particularly, which is a DISA family. These proteins contain an amino terminal helicase domain and several other domains, carboxy terminal segment, and so on. And all these domains would recognize and cleave are used for different domains, are used for different functions to cleave the double-stranded RNA molecule. DISA family proteins activity is ATP dependent. These are nucleases because they have to cleave the double-strand RNA. They also contain the other domains like PAS, which is thought to be important for protein-protein interaction. Loss of DISA means loss of silencing. RISC complex. This is RNA-induced silencing complex, which we discussed just before. It's a large, more than 500 kilodalton RNA multi-protein complex, which triggers mRNA degradation in response to siRNA, double-stranded siRNA. Unwinding of double-stranded siRNA by helicase, one of the enzyme activities of RISC complex, and ribonuclease component cleaves mRNA nuclease, which is a nuclease activity. And amplification of silencing signal, RNA-dependent RNA polymerase, will increase the RNAi, and this would cleave target mRNA, and this double-stranded mRNA is degraded by other cellular exonucleases. Different class of small RNA molecules. You should know the terminology here, particularly when you're talking about the gene silencing. siRNA is known as small interference RNA, which we talked about, and miRNA is a microRNA, and another is stRNA, small temporal RNA. All these RNAs are small nucleotide sequences, 21 to 25, 26 base pairs. These RNA sequences, they are single-stranded when they are produced. They go and anneal the complementary sequence of the target transcript, thereby making the transcript susceptible for cleavage by different enzyme systems. What are small interfering RNAs? Small interfering RNAs that have a target role in the phenomena of RNA interference, a form of post-transcriptional gene silencing. RNAi, they are of about 21 to 25 nucleotide long fragments, which bind to complementary portion of the target mRNA and tag it for degradation. A single base pair difference between siRNA template and the target mRNA is enough to block the process. MicroRNAs or sTRNAs, they are small, again, molecules, 21 to 25 bases long, and they derive from approximately 17 nucleotide single-stranded RNA, which form stem loop structures, and these stem loop structures are processed to produce 22 nucleotides, approximately to 25. sTRNAs do not trigger mRNA degradation. They are just temporal. Why is post-transcription gene silencing is important? Most widely held view is that RNA evolved to protect the plant against the viral infection. The viral RNA, when it's transcribed, and this RNA need to be neutralized, so the microRNA which are produced by the plants can complement with the viral RNA, form a double-stranded structure which gets subjected to degradation. And recently, these microRNAs or siRNAs, which have been discovered in eukaryotes, they do a lot of functions. They regulate large RNAs and their expression and their silencing, thereby impacting the development of the organism. This RNA technology is utilized to study the gene functions. So students, let us summarize this module. Today I tried to explain the features of various selection and reporter gene markers and their usefulness in identifying transgenic plants and the differing genetic and physiological processes in plants. Further, we also analyzed the strategies employed to silence gene expression in plants and their utility in understanding gene function and cellular processes. Our discussion also analyzed as to how the knowledge so acquired on gene functions can be utilized to modify the metabolic pathways and incorporate the genetic networks to enhance plant performance. Thank you.